Hello everyone. I welcome you at the Advanced Multiple YouTube channel. Advanced Multiple Incorporation actually a knowledge processing and information management organization that is located in Mississauga, Ontario, Canada. My name is Dr. Taha Nazir. I'm working as a scientific executive with the Intellectual Consortium of Drug Discovery and Technology Development Incorporation. Both of these organizations are located in Mississauga, Ontario, Canada. Okay, well, today I'm going to share my information, knowledge, and my experience under the title of Epidemiology and Characterization of Drug Resistance Organisms around the world. For this purpose, I collected the information from different data sources like PubMed, Medline, Scopus, Google Scholar, National Library of Medicine, MBAS, Directory of Open Access Journal, ProQuest, CNKI, that is stand for Chinese National Knowledge Infrastructure, that is China-based database company that provide the information scientific research all over the world. Some information collected from Crossref and a ResearchGate. Okay, before moving forward, I would like to talk a little bit about epidemiology, the study and analysis of the distribution pattern and determinant of health and disease condition in defined population or in particular people called as epidemiology. It is a cornerstone of public health worker to design policy, take decision, and practice on basis of evidence to identify the risk and set the targets for to prevent the disease. Epidemiology is the study of how often disease occur in different group of the people and why. Epidemiological information used to plan and evaluate strategies to prevent illness and guide the management to handle the patient and disease. Okay, well, there are different kind of the microbial organism. They are resistant to different kind of antibiotic used in clinical setting in hospitals. Antimicrobial resistant render the effectiveness of medication used for the cure for the treatment of the infection. Some of the resistant strain may be referred as superbugs. These superbugs are very resistant. They are the big challenge for the healthcare professional and clinician. And we try different kind of the combination to make sure the treatment of infection. So these resistant infection may kill and can spread to other and impose the huge cost of individual on and on society. The potential superbugs for the resistant organism are multi-drug resistant tuberculosis, methicillin resistant staphylococcus aureus, New Delhi metallo-meta-lactamase 1 strain, car- carbapenem resistant enterobacteriaceae, vancomycin resistant enterococcus. These are the resistant strains around the world identified in different institutions. So these are the potential challenges for the clinician, for healthcare workers. Okay, well, this is the brief historical review of the bacterial resistance, and this is a brief history of the worldwide first superbugs. In 1928, one of the scientists named Alexander Fleming, he was working in the laboratory. He observed that one of the mold, Penicillium notatum, he produced some of the chemical that kill the bacteria. Bacteria means staphylococcus. So that chemical produced by the mold eventually named with the penicillin. So Alexander Fleming was the first person who discovered the first antibiotic to kill the bacteria in the world in human history. In 1946, the penicillin was commercially launched for the utilization in the clinical setting and in hospital. Second World War, a lot of the injured patients were saved because of this penicillin because it is very frequently and sufficiently widely used for the soldiers injured during the war. In 1947, we see the resistance against the penicillin. So 1959, there is a first synthetic penicillin with the name of the methicillin was introduced in the market, and that was able to kill the resistant microorganism like Staphylococcus. In 1961, the first Staphylococcus aureus he was noticed and identified in British hospital, and he was resistant to the methicillin. This was the strain afterward called as methicillin resistant Staphylococcus aureus. This is a very typical class that is still prevail around the world. So in late 60, more of 80% of the community and hospital acquired patients having the resistance against the penicillin. So this is the very basic, very fundamental history and historical review of bacterial resistance. Okay, well, now I would like to talk a little bit about resistance against the antibiotics. There are the four different mechanisms by which the resistance develop in the bacteria. Obviously, there are many other mechanisms, but there are the four major mechanisms by which bacteria produce the resistance against the antibiotics. The first one is the inactivation of the drug by the enzyme. Bacteria produce some enzyme that deteriorate, destroy, or damage the drug. When the drug is damaged, then obviously it is not available in the patient body. It is inactivated, destroyed, and damaged, and that may not able to produce the pharmacological effect and kill the bacteria. Second mechanism by which the resistance produced in the bacteria is the alteration of the drug target, modification of the drug target. All antibiotics, they have their particular targets on the bacterial part, bacterial segments or the bacterial body. So some of the antibiotics, they attack on the cell wall. Some of the antibiotics, they attack on the cell membrane. Then some of the antibiotics, they attack on the proteins of the bacteria. It may be the ribosome, maybe the nucleotide, maybe the chromosome, maybe the RNA. So different antibiotics having the different targets where they attach and they kill the microorganism or the bacteria. If the target is modified, its chemical nature is changed, its chemistry is changed, then the drug is unable to attach in his target or in a particular domain. So bacteria become resistant. The third mechanism by which resistance develops is the change in the chemistry of the cell wall. If the chemistry of the cell wall is changed, then drug is unable to penetrate or enter into the cell of the bacteria. If it is unable to enter into the bacterial cell, it means it is unable to work. It is unable to kill the bacteria. If it is unable to kill the bacteria, then infection will remain and produce more and more complication in the patient, and eventually may produce very serious consequences, and bacteria eventually become resistant. The fourth and final mechanism by which resistance is developed in the bacteria is the activation of the drug efflux pump. There are some pump, they excrete or pump out the toxins or the chemicals or the drug from cytoplasm to outside the bacterial cell. When drug is pumped out, that is effluxed or excreted from the body, then they may not produce the effect. They may not kill the bacteria. When bacteria is not killed, then it means we are unable to control the infection. So these are the four mechanisms by which most of the time resistance produced or developed in the bacteria. Okay, but there are certain facts regarding the antimicrobial resistance. The poor quality medicines exposing patient to suboptimal concentration and the poor access create the condition for the drug resistance to develop. Subtherapeutical dose in animal for promoting growth or preventing disease can result in the resistance which can spread to human. Poor infection control can increase the spread of the drug resistant infection. Hospitalized patients are one of the main reservoir. They were carrier of potential resource of resistant microorganisms. Currently, only few established network collect and they report the resistant data on regular basis that impair the ability to detect emergence of the resistant microorganism and take prompt and quick action. Existing antimicrobials are gradually losing their pharmacological effect. So WHO called for action on the World Health Day to halt the spread of the drug resistance by introducing 6-point policy package all over the world. This is the illustration of the plasmid. The plasmid is one of the genetic material most of the bacteria having in their cell. This plasmid having competency to compete or the establish the or produce the resistance against the drug. A plasmid might have the resistance against multiple drugs. This plasmid having the resistance against 10 different antibiotics, including tetracycline, trimethoprim, penicillin, family resistant, and vancomycin. Okay, well, This diagram actually highlights the site of suspected or confirmed artemisinin resistant in Greater Mekong subregion during 2006 to 2013. So this study was carried at about 7 years. They collected the information and compiled and published. So Great Mekong subregion comprised of the Myanmar, Laos, Vietnam, China, Thailand, and Cambodia. Okay, well, this diagram actually highlight or represent the percentage of the new tuberculosis cases with the multi-drug resistant tuberculosis. We know there are the three main kind of the tuberculosis patient. First one, the susceptible tuberculosis. They have the mycobacterium tuberculosis. They are susceptible to the antibiotics. Antibiotic means first line therapy. First line therapy means rifampicin, isoniazid, ethambutol, and maybe with the streptomycin or without streptomycin. The second type of the tuberculosis case is having the multi-drug resistance, and multi-drug resistant are the mycobacterium tuberculosis, which are at least resistant against two antibiotics, including isoniazid and rifampicin. So the third kind of the patient are the extensive drug resistant. They are the more serious, more dangerous patient, and this kind of the mycobacterium tuberculosis, they are almost resistant to all available antibiotics. The darker area that indicate the higher percentage of the tuberculosis cases, and the dark area in this diagram actually indicate the higher number of the patient or higher population of multi-drug resistant tuberculosis. The lighter area or the light color area, they indicate the low percentage or low number of the patient of multi-drug resistant tuberculosis. So we can see that in Russia, China, and Central Asia, they have a higher population. They have higher number of the patient with multi-drug resistant, and the like America, then Canada, Mexico, and South America, African countries, Australia, they have low percentage of the patient with multi-drug resistant tuberculosis. Okay, well, this is the diagram that actually indicate the percentage of the patient with previously treated tuberculosis cases with multi-drug resistant tuberculosis. So the patients, they are treated successfully, but after a while, maybe after 1 year or maybe after 6 years, they relapse. They become infected again with the multi-drug resistant tuberculosis. So the darker area, similarly, they indicate the higher number or higher population of the patient with the multi-drug resistant tuberculosis, and the lighter area, they actually indicate the lower quantity, lower number of the patient with the relapse multi-drug resistant tuberculosis patient. Okay, well, in precautionary measure, we should have better hygienic condition. We should have access to clean water and sanitation. Infection control and healthcare facility should be available. People should be vaccinated properly. We should have to establish compliance successfully. Rational antibiotic usage, and use antibiotic only when prescribed by the healthcare professional or the physician. Complete the full prescription, full treatment protocol, even you feel better. Never share antibiotic with other or use leftover prescription without prompt action. We may move back to post antibiotic era in which common infection or the minor injuries were became more serious or the mortal. So we should be careful and vigilant. In conclusion, antibiotic treatment of infectious disease may potentially be modified to cope with the pattern of the drug resistance. Development of the new antimicrobial agents and increasing frequency of multi-drug resistance mean that we need more effort to ensure the effectiveness and give chemotherapeutic agent in more rational manner. Future development toward the clinical and therapeutical application will likely combine an existing therapies with new scientific techniques. Okay, well, at last, I would like to greet and acknowledge the contribution and support of my team, my colleague, and my friends like Shiri Bajkar, Bulk Gang Dang, Daniel, Dustin Hawker, Marwa Al Maghribi, then Robert, then Amin, Unza, Noreen, Arslan, Ishtiaq, and Terry Mill. I would like to acknowledge the participation, support, and help of Advanced Multiple Incorporation and Intellectual Consortium of Drug Discovery and Technology Development Incorporation. As I told you in the start of the video, both of these organizations are located in Mississauga, Ontario, Canada. Thank you very much for watching the video and staying with me for so long time. And see you soon in my next video. Take care. Bye now.